Dear students, in the previous lectures on the extracellular matrix and tissue biochemistry, you have extensively seen the details of structure, functions, biomedical significance, biosynthesis, and degradation of collagen and elastin. You have also done in detail collagenopathies. So today we are going to start genetic disorders associated with elastin, and we are also going to focus on two special proteins of extracellular matrix, which are fibronectin and laminin. Aims and learning objectives of today's lecture include to study in detail genetic disorders associated with elastin, like Williams-Burden syndrome, supravalvular aortic stenosis, and one indirect disorder associated with elastin, that is Marfan syndrome. And then we'll emphasize upon two special proteins of extracellular matrix, that is fibronectin and laminin. The very first genetic disorder that we are going to study associated with elastin is Williams-Burden syndrome. It is a kind of developmental disorder that affects connective tissue and central nervous system. Elastin gene, which is located at chromosome number 7, has been reported to undergo deletions, and as a result of which it becomes defective and may cause this disease. In 90% of individuals affected with this disease carry this type of mutation in elastin gene. Supravalvular aortic stenosis is a type of defect or abnormality in the heart that is developed prior to birth. It is characterized by stenosis or narrowing of the large blood vessels, which are responsible to carry blood from the heart and distribute it to the rest of the body. It is thought that synthesis of elastin is defective in this case, and this is due to mutations in the elastin gene. For example, elastigene or ELN gene that gives instructions for the formation of tropoelastin has been seen to be mutated or abnormal in this case. Williams-Burden syndrome patients are most oftenly presented with this defect. Elastin may undergo fragmentation, and thus it becomes decreased in the conditions like pulmonary emphysema that we have studied in detail in the last lecture under the heading alpha-1 antitrypsin deficiency, and also in the process of aging of the skin. Now the next disorder that we are going to study is Marfan syndrome. Marfan syndrome is also a genetic disorder that affects connective tissue. In this disorder, there is defective Fibrin 1 protein. Fibrin 1 protein is a type of a large glycoprotein. It is found in lens, periosteum, which is the outer covering of the bones, and aorta. And even it is found elsewhere in the body. It is basically a key structural component of microfibers. It is first synthesized in the fibroblast, and then it is secreted into extracellular matrix. Then it becomes incorporated into microfibrils. Now, microfibrils basically give support or scaffold for deposition of elastin. So it means that if fibrin 1 is defective, then elastin cannot be supported with the microfibrils. So Marfan syndrome, defective Fibrin 1 affects eyes, skeletal system, and cardiovascular system. In eyes, it basically causes ectopia lentis, which is basically the dislocation of the lens. And then in skeletal system, the defective cause is called arachnodactyly, in which most patients appear tall and they show long digits, and there are also hyperextensible joints. The third defect is seen in cardiovascular system. It basically causes the weakness of the aortic media, which is the middlemost layer of aorta, and as a result of which it leads to the dilation of the ascending aorta. So this is ectopia lentis, in which you can see displaced lens. This is arachnodactyly. And this is the ascending aorta that may be found dilated in Marfan syndrome. Marfan syndrome is caused by mutations in Fibrin 1 gene, and the gene for Fibrin 1 is found on chromosome number 15. Missense mutations have been reported in several patients of Marfan syndrome. As a result of these mutations, there is the formation of abnormal fibrillin, or lower or less amount of fibrillin is available to be deposited in the extracellular matrix. So abnormal fibrillin, or when it is present in less amount in extracellular matrix, then it cannot support elastin, and hence connective tissue disorders or connective tissue abnormalities develop. So the next protein which is included in your syllabus is fibronectin. So we are going to see biochemical aspects of fibronectin, and especially we are going to focus on its significance or role in cellular adhesion and migration. Now first of all, location of fibronectin. Fibronectin is found as a major glycoprotein in the extracellular matrix, and it is also found in blood plasma. Now what is its structure? It basically comprises two subunits which are identical, and these are joined together by two disulfide bridges, and these disulfide bridges are found near their carboxyl or C terminals. The structure of fibronectin is further distributed into functional domains. For example, you can see that this is the fibronectin structure. There are two identical chains, and at the C terminus you can see disulfide bonds. And these are all the functional domains of fibronectin. The domains in the fibronectin are for binding fibrin, for example, for binding heparin, for binding cell surface, again binding with fibrin, to bind with collagen, and then to bind with heparin and fibrin. There is a special sequence found in fibronectin which is called RGD that we are going to see later on. So the functions of the domains of fibronectin include its binding with heparin and fibrin, with collagen, even it can bind with DNA and with the cell surfaces. There are receptors on the cells which can bind to fibronectin, and these receptors are simply called as fibronectin receptors, and these may also be referred as integrins. So these are present on fibroblasts. These are transmembrane receptors, which means that they can pass along the membrane. These are heterodimers, which means that these are composed of different subunits. So they may contain different types of alpha and beta polypeptide chains. Fibronectin contains a special sequence which is called as RGD. RGD stands for arginine, glycine, and aspartate. These are the amino acids. Now this special sequence basically binds to the receptor. Overall, fibronectin does not bind with the receptor. Fibronectin binds to its receptor with the help of this special sequence which is called RGD. So here you can see that this is the RGD sequence, and through this sequence fibronectin can bind to its receptors which are called integrins. So this is the figure which is showing fibronectin receptor, which is also called as integrin. It is composed of two domains A and B, and here this integrin receptor is making association with fibronectin, and this is that RGD sequence through which fibronectin is binding to its receptor. Other types of fibronectin receptors can also bind directly with collagen, and they can also bind to another protein called laminin. This is another figure which is showing the binding of fibronectin with fibronectin receptor. Now this is the fibronectin receptor, which is also called integrin receptor. It is composed of two subunits A and B, and this is the fibronectin protein. Now this fibronectin protein is bound to the receptor through its RGD sequence, and the functional domains of fibronectin can bind to other molecules as well. For example, it can bind to collagen, it can bind to heparin, it may bind to fibrin and others. Now this is the outside of the plasma membrane, which is called as extracellular matrix. Now towards the inside of the cell, fibronectin receptor can also bind to a set of attachment proteins. For example, talin, vinculin, capping protein, and alpha actin, and these proteins then make binding with actin. So basically fibronectin receptor is sandwiched between fibronectin and attachment proteins. Fibronectin is found in extracellular matrix, while attachment proteins are found inside of the cell. When the fibronectin interacts with its receptor, then basically it promotes the communication of the exterior of the cell with the interior of the cell. Thus, overall, it affects the cellular behavior. Fibronectin plays important role in the cellular adhesion, and with the help of fibronectin, one cell can adhere to another cell, and thus it is also involved in cellular migration. Laminin is the last protein of extracellular matrix that we are going to study. It's a protein that comprises three elongated polypeptide chains, namely A, B1, and B2. These chains are joined together in the cruciform shape, which is basically a cross-like shape or crisscross shape. Laminin provides binding sites for type 4 collagen, heparin, and integrins. Entactin is another protein that can bind to laminin. It is a glycoprotein which is found in the basement membrane. It also contains a special sequence called RGD, as we have seen in the case of fibronectin. Basal lamina or basement membrane contain laminin protein. So here we are going to see functions of basal lamina of the renal glomerulus. So in kidney glomerulus, basal lamina acts as a permeability barrier, and it also acts as sieve and glomerular filter, which means that it regulates the trafficking or passage of large molecules, for example proteins, across the glomerulus and then into the renal tubules. So basically, basal lamina of the renal glomerulus is used to filter large molecules, for example large proteins, and thus these cannot be secreted into the urine. Glomerular membranes can only allow small molecules like water, inulin, and some others to pass through. Large particles, large proteins, large molecules cannot pass glomerular membranes, and therefore these are fully filtered. But there is one question, that albumin is as small as water, but it cannot pass glomerular membrane. What is the reason behind it? This is because there are negative charges which are contributed by heparin sulfate and some other proteins in the basal lamina. And therefore these negative charges can repel negatively charged proteins, for example albumin, at the pH of the blood. So it means that albumin, though small like water, but it cannot pass glomerular membrane because this is negatively charged and glomerular membrane is also negatively charged. So both of these repel each other. But when the glomerular membrane is damaged or the structure of glomerulus undergoes damage, for example, in these conditions like glomerulonephritis, which is the inflammation of the glomerular membrane, or the nephrotic syndrome, which is a kind of kidney disorder, or diabetic nephropathy, this is again a kidney disorder due to diabetes. So in all of these conditions and other kidney related conditions in which renal glomerulus is damaged, there is release of albumin. So albumin can appear in the urine of these patients. So the condition in which albumin appears in the urine is called albuminuria, and this is a type of proteinuria.